Welcome, everyone. I'm not going to do the introduction today. I'm just going to say the line that you all know, and then I'm just going to pass it to Anastasia Yendiki, yes, who will basically do the introduction. But welcome, everyone. And as you all know, this seminar is co-sponsored by the P41, and I can't still say it, the P41 Center for Mesoscale Imaging House in Martinos. But this seminar specifically, we are also co-sponsored by the Center for Large-Scale Imaging of Neural Circuits, or LINC, which I'm sure Anastasia will tell us more about, or not. But then I'll just hand it over to Anastasia to do the intro. So thank you very much, everyone.
So special guest today's Professor McIntyre is coming to us from Duke University. He's a professor of biomedical engineering and neurosurgery there. He got both his bachelor's and PhD. Oh, sorry. To the hot mic, right? He got both his bachelor's and PhD from Case Western Reserve University. I'll skip the dates so that we don't date you. His doctoral research focused on the biophysics of neural stimulation with implanted electrodes. Then he did his postdoc at Johns Hopkins. And then he started a research group at Cleveland Clinic. Then he was back at Case Western for a while and then moved to Duke. And the special expertise of his group is in the theoretical, experimental, and clinical application of neuromodulation and neurorecording technologies in human patients. And with that, I'll hand it over to Cameron. Thank you.
All right, great. Thanks for the invitation to be here. I think I have been here before. It was about 10 years ago now, so it's great to have a chance to come back and learn about all the cool imaging stuff that you guys are doing. And yeah, so today I'm going to talk about some stuff that I think is relevant to this new LINC project and how some of these kind of modeling strategies that we've used in the past might be applicable or useful as we start moving forward with kind of this next generation of brain connectomic modeling. Okay, so my kind of research group has been almost exclusively focused on what I would call deep brain stimulation biophysical modeling for quite some time, and I break that down into kind of two different categories of modeling. So when we think about deep brain stimulation, you think about stimulation. So you're applying an electric field from an electrode. That electric field is polarizing a neuron, and then we want to understand how the neurons respond to the stimulation. And then the other side of the equation, and especially relevant now in the world of clinical deep brain stimulation, is the recording side. So now there are many devices, commercial clinical devices out there in the world that are doing local field potential recordings. We want to understand what is the biophysics of the neural firing that then leads to these kinds of local field potential recordings. So I'm going to try and give you a flavor of both of those things. And both of those things are part of this kind of bigger context of kind of network modulation, which is the primary purpose of these deep brain stimulation therapies. So we use deep brain stimulation therapy in all kinds of different disorders, but the most common and the most widely used is deep brain stimulation for Parkinson's disease. The most common surgical target for that is the subthalamic nucleus here, which is part of this kind of thalamocortical basal ganglia, kind of loop, and through kind of clinical trial and error, people kind of came to this realization that this was a great place to put our stimulation electrodes to then modulate this bigger network. But always keep in mind, there are many different circuits within this network, and so when we stimulate in one place, for example, this kind of little part of this network, you'll have reverberating implications throughout, other parts of this network. And that's kind of like the hot topic right now in deep brain stimulation research is trying to understand how these different networks are directly influenced by the stimulation. How can we record from different parts of this circuit to understand that modulation? And then it would be great if we could, kind of turn that problem on its head and come up with better, more efficient, more effective strategies to modulate that network in the most efficient or effective way possible. So, so one thing that has come out of DBS research over the last, I would give it about a 15-year run, where this one part of this circuit has become extremely interesting and exciting to many, many people. And that is this component that is known as the hyperdirect pathway. So these are layer 5 pyramidal neurons that have an axon that projects down to the subthalamic nucleus, and it is this direct cortical excitatory drive into the subthalamic nucleus that people have kind of honed in on as being a key element of, kind of the therapeutic mechanisms of deep brain stimulation. So I'm going to, kind of go through, kind of an evolution of how did we come to that understanding, or what can we, kind of glean from that sort of basic science evolution into something that is relevant to clinical analysis. Okay, so where are our first step, and that is going to be this kind of looking at this concept of DBS local field potentials. So that became a really hot topic in the field of deep brain stimulation. I give that about a 20-year run or so. So about 20 years ago, people would be doing intraoperative electrophysiology experiments in patients, and they were noticing these really robust beta band, local field potential recordings with a really prominent, kind of beta frequency to them in the subthalamic nucleus of deep brain stimulation patients. And they also noticed at that time that when you gave drugs to these patients, or when you turned on the stimulator, dopamine replacement type drugs, these beta band oscillations would go away, and then they would kind of gradually come back as the patient would, kind of go back to having Parkinsonian symptoms. And so the idea that this was like an electrophysiological biomarker that you could record from and use as a trigger for turning stimulation on and off. So that's kind of what's depicted in this picture down here. You might have this kind of frequency, kind of spectrum that you're recording from. If the power in a certain frequency band gets above a threshold, then that's a turn on or trigger to turn on your stimulation. And this became a really hot topic in deep brain stimulation research. Like I said, okay, now that would be like about 15 years ago, people got hot on this idea that you could do closed loop, electrophysiologically triggered clinical deep brain stimulation for Parkinson's disease. Then about 10 years ago, clinicians started having devices that they could prototype that kind of thing. And then 5 years ago, that became kind of widespread. And then just like last year, you have like commercial grade devices that people can do this with in real time in real patients. Okay, so the time constant of, kind of application of a scientific idea into a clinical product is so long, like most scientists cannot possibly understand how long that is for a super simple idea even, right? So anyway, all right. But here was, kind of part of that step, right? Is this idea that this is a frequency spectrum recorded from a deep brain stimulation electrode of a Parkinson's patient in the subthalamic nucleus. This is the, kind of the power in these different frequency bands in the PD state. So in a normal brain, you would kind of have this 1/f power law that is just sort of like the, kind of baseline neuroactivity. But in a Parkinson's patient, you've got this hump of exaggerated activity at approximately 20 hertz. If you give that patient subtherapeutic DBS, so you turn on the stimulator, but you don't turn it up very high, that beta band, kind of peak doesn't really go away. But if you turn up the stimulation enough, boom, that goes away. And that correlates with, improvement in symptoms. So this idea, got a lot of traction. People were very excited about it. But I've always been interested in, well, what is this signal? Where does this signal come from? What is creating it? And so we got these local field potentials, right, that are being recorded from these macro electrodes. So these are 1.5 millimeters in height and about 1 millimeter in diameter. And so, you're getting these macro recordings. And like I said, they have a clinical relevance to them as a biomarker. But, the field wants to understand, like, well, what is creating these waves, right? So the waves are coming from the firing of individual neurons and their synaptic inputs. And you can line that up and see that, like, oh, when the neurons are firing, you get these, kind of negative deflections. When the neurons aren't firing, you kind of get these positive deflections and so on and so forth, right? So you could do that in the operating room. You could record from individual units, right, with a microelectrode next to your deep brain stimulation electrode. You could get kind of this, lining things up, but we're not able to, really answer the questions of, like, well, how many synchronous neurons are we talking about? Are we talking about 10? Are we talking about 10,000? Are we talking about 100,000, right? I mean, to me, that's kind of an interesting science question. And then where is that synchrony located? Is it all the entire nucleus, everything firing in synchrony, or is it just small parts of the nucleus that are creating this pathological biomarker, if you will? So you can't answer that kind of question with experimental electrophysiology, especially not in a human. So one of the ideas that we had was, well, what if we use these kinds of anatomically inspired, biophysically detailed neuron models to get at that? And so the first step of that process is you need to have a model of an individual subthalamic nucleus neuron, right? So here's a 3D reconstruction of a non-human primate STN neuron. You can then break, right, here's the cell body, the dendrites, this is the axon coming out there. You can then break that neuron up into a whole bunch of little tiny electrical circuits and make what we would call a multi-compartment cable model of a neuron, where each compartment then has its own little complement of sodium, potassium, calcium channels that are responsible for the kind of action potential signaling that a neuron, exhibits. And then, the cool thing is that then now you've got a quantitative metric for all these transmembrane currents that are then subsequently creating these field potentials that we can record from these electrodes, right? So these neurons are being driven by synaptic inputs. Interestingly, these kinds of neurons have, kind of predominantly inhibitory inputs hitting their cell bodies and proximal dendrites, and then they get predominantly excitatory inputs out on their distal dendrites. And those two competing synaptic drives are, obviously modulating the firing of the neuron and then creating these kinds of little current sources all over space around that neuron, right? And then those current sources are going to sum up and create then something that we can record on these big macro electrodes. So now if you've got an idea or you've got a simulation that can give you an idea of where these currents are coming from, then we can start to think about, well, let's put that in the context of an individual patient brain. So our group has a long history of kind of using, kind of clinical MR data sets to then create, these, let's call them patient-specific anatomical models of the thalamus here in yellow, subthalamic nucleus in green, and then, kind of the estimated placement of the deep brain stimulation electrode in that particular patient. So that would give us now some kind of a model of the relative anatomy around our DBS electrode. And then you could think about, well, each little voxel, if you will, of that subthalamic nucleus, well, I could put a little STN neuron model there. And then I could have ideas of the currents that are being generated by each one of those little neurons, and then start to back calculate what would be the local field potential recording I might expect to get at these different electrode contacts, right? So a clinical, old school clinical deep brain stimulation electrode would have 4 contacts on it. Nowadays, the modern electrodes have 8 contacts. And in fact, actually later this year, we'll have 16 contact DBS electrodes. So, things keep getting more complicated, but if we now, kind of start putting this together with, kind of these pink contacts being our electrode, and then here's, a variety of our neurons distributed in space around it. And then, well, what if we kind of, put 1% of the density of real neurons in the STN? Like now you can barely even see the electrode. And well, what about if we put the real density of neurons in there? You have about 250,000 neurons in your STN. Now you have this super, super densely packed population of neurons surrounding your electrode. And the cool thing about computer models is like, well, we can make 250,000 of these neurons if you got a big enough computer. There's nothing to stop you. So imagine now at each one of these dots, we've got one of these, kind of little multi-compartment neuron models that can generate these current sources, just like we think real neurons do. So then the next step in that process is, okay, now we got our 250,000 neurons distributed with anatomical realism, if you will, around our DBS electrode. And what creates a local field potential? What creates a local field potential is synchronous neural activity firing in near proximity to the implanted electrode contacts. And so now we can control this simulation where we're going to say, okay, all those green neurons, we're going to make them synchronous. We're going to make them synchronous in 20 hertz, kind of oscillatory patterns of activity. And then all the rest of the neurons in blue, they're just going to be kind of asynchronously firing. They're going to be firing like real neurons do, but they're not necessarily synchronous with each other, right? So how do we do that? We drive the synaptic inputs to those populations of neurons differently, such that we create transmembrane currents in them that are either really parallel with each other, or driving, firing in synchrony, or they're firing, basically is like background noise, right? So if we do that, then we can start to create now recordings, simulated recordings on the DBS electrodes that correspond with, either different, recording setups that you might do in the kind of clinical operating room or in the patient chronically. And then you can now match that up with the simulated recordings that you create in your model compared to clinical electrophysiological recordings that you get from this exact same patient with this exact same electrode location and so on and so forth, right? So this is what we, kind of try to do. We start building these models, we build these simulations, try and compare it to the actual electrophysiology of the patient. And one of the questions that we wanted to look at was, well, what happens to the signal if we move this kind of synchrony around, right? So we didn't think that the whole STN was going to be synchronous at once. That wouldn't really make much sense. We know a lot about how the STN is kind of broken up into different compartments or different regions. And so, as you can imagine, as your signal, your local field potential kind of signal amplitude changes as you change kind of the position of the synchronous volume, right? So as the synchronous volume kind of moves around the STN, you're going to record different, kind of signal amplitudes at the different electrode contacts, right? And part of the thing that is kind of cool about these modern devices is that you can record parallel, simultaneous activity in many, many different contacts, right? That's the whole point, is that it gives us an idea to triangulate or localize, where is this activity coming from? So if we do that on, kind of through a big search space, right, we can look at, kind of the radius of that synchrony. We can look at the position of that synchrony in the subthalamic nucleus. And then you compare that up against the clinical recordings, right? So you do kind of like a match between the recordings, the bipolar recordings at channel 0, 1 compared to channel 1, 2, so on and so forth. And you try and find, kind of where does the model create a best fit to the actual clinical recordings from that same patient. And you run through all this, and I'm sure no one cares about the math, but the cool kind of output of this is that for the first time, we kind of give ourselves an estimate of like, all right, to get the signals that we're seeing in these patients, we'd say like probably going to be needing about 35,000 synchronous neurons, and those neurons are probably going to be located up here in this dorsolateral subthalamic nucleus. Okay, so that was kind of a really cool thing because that's also lines up perfectly with the anatomy, right? And getting back to, where do these things start to converge? So this was a beautiful piece of work that Suzanne Haber's group had done a few years earlier, where they were now taking micro injections of tracer in the cortex of non-human primate macaques, and then looking at where that, those hyperdirect axon terminals were ending up in the subthalamic nucleus. So if you put the tracer in M1, those kind of like synaptic inputs would terminate in this dorsolateral STN. If you put the, the tracer in the prefrontal cortex, you would get these tracers, or you would get the synaptic terminals ending up in the kind of the anterior subthalamic nucleus, right? And so it would make sense that if we've got a movement disorder that's related to, kind of dysfunction of the motor system, then if we've got abnormal, kind of pathological neurophysiology activity is probably coming from the motor system, which is then corresponding with this hyperdirect innervation to the subthalamic nucleus. And that's what the model simulations or the biophysics would tell us as well. So that's kind of like one piece of data that's like, oh yeah, this hyperdirect pathway seems like it's really got an important role to play in DBS. Yeah.
Quick question. So these neurons, you did not connect them to each other, right?
No.
The only thing was the input, which.
That's right. That's right. So we're artificially creating the synchrony as opposed to letting a network fall into a synchronous pattern, right? That's different kind of network modeling.
Right.
But in this case, we wanted to explicitly control it ourselves, and that's why we set it up that way. Yeah. Okay, so at that same time, once again, now we're kind of, take yourself back to kind of, I don't know, 2015 time window. People are now have recognized this concept anatomically of the hyperdirect pathway. They've recognized this concept of, abnormal beta band oscillatory activity in the subthalamic nucleus as being a biomarker of Parkinson's disease. And then on top of that, people are doing simultaneous recordings up in cortex, seeing that same beta activity. And with all these pieces together, coming to this conclusion that there is most likely a strong cortical beta drive that, is coming from, these higher inputs and feeding itself into the basal ganglia, maybe primarily through this hyperdirect pathway. And so we've got this kind of issue of, we've got recordings and physiology telling us something. How does that line up with the stimulation side of deep brain stimulation, right? And so now we'll kind of move to, well, how can we start to characterize the effects of stimulation using these kinds of techniques? So the primary mechanism or tool that our group has used in that process is what we would call these patient-specific deep brain stimulation models. And so this idea that you've got, kind of highly detailed MRI-based models of individual patients. We zoom into this, kind of subthalamic nucleus region again. And this is, kind of an example patient where like the electrode is in a perfect place. So we would say like, we know there's like a clinical therapeutic sweet spot that's in the dorsal lateral posterior kind of STN, right on the border of, kind of the dorsal border of the nucleus. So that red contact. And we've known that for a long time, like that's not new. But we've also never really known, well, what is it that we're stimulating in that region? Because there's a lot of stuff there. So one thing that, people used early on to try and use as a proxy to understand stimulation is, we don't know all the parts of the basal ganglia that are, going to be stimulating in that area, but for sure we know that there's a gigantic white matter superhighway running right by the electrode called the internal capsule, right? And it's something that can be easily reconstructed with tractography on a patient-specific scale with a good deal of accuracy. So we can build, kind of these streamlined models of this pathway, the internal capsule, as it courses by our deep brain stimulation electrode. And then now we, instead of doing the recording side of the DBS electrode, we do the stimulation side. So now we're imposing an extracellular electric field into the tissue medium. That creates a voltage distribution along all these different axon pathways, right? Or individual fiber models. And then we can kind of pull one of those, kind of axons out of that mix, and then just like kind of dig down a little deeper into what happens when we polarize it with this electric field. So the deep brain stimulation electric pulse from a clinical device looks like this. It's kind of a short duration, high amplitude, kind of 90 microsecond, 60 microsecond pulse, followed by this very long duration, low amplitude kind of recharge phase. So this would be the cathodic phase of stimulation. Then there's an anodic phase of stimulation that follows it. And that is something that can be polarized or modeled. The polarization of the neuron can be modeled, once again, using these same kinds of Hodgkin-Huxley style multi-compartment cable model representations. So once again, we've got our sodium, potassium channels that are going to be responsible for our action potential signaling. They're going to have an electric field applied out here. So this is the extracellular space where I'm pointing right here. So that extracellular voltage distribution is going to get applied to that. That's then going to cause currents to flow through this circuit and then eventually create, the polarization of the neuron and subsequently the creation of action potentials. So when you generate, kind of an extracellular stimulus, the action potential initiation from a cathodic pulse is going to occur in that node of Rambier that's, kind of closest to the electrode. So boom, the action potential starts, and then it's going to propagate in both directions along the trajectory of that fiber, right? And so kind of the point and purpose of these stimulation therapies is that we're hijacking the neural firing of the neurons around the electrode to now do what we want them to do. You're firing along, doing your own thing, not anymore. We're taking over. We're controlling your firing now. And so we can then use this concept of what we call a pathway activation model to then come up with these estimates of pathway recruitment as a function of our stimulation parameter settings, right? So as our stimulus grows, the voltage distribution, kind of amplitudes grow in space. And as those voltage distributions kind of get stronger and stronger, they start to activate, different axons within that population. And you can get to the point where you've got kind of an estimate of stimulus activation in different pathways. And so that example was obviously from stimulating the internal capsule. We don't want to stimulate the internal capsule in real life. We're trying to avoid it. It's the big side effect generating pathway around these clinical electrodes. We're actually trying to avoid stimulating that. But the things that we do want to stimulate from a therapeutics perspective, like we have barely any understanding of what they look like anatomically, right? So these are like anatomist drawings from the 60s and 70s that tell us that there are these pathways that connect the globus pallidus, here in blue, to the thalamus, and the subthalamic nucleus, here in yellow, to the globus pallidus, and so on and so forth. But like at least 15 years ago, like we didn't have like a realistic representation of what these things looked like, right? And so to do the kind of stuff that our lab wants to do, we need a picture more like the one on the right there. Like we need a three-dimensional anatomical representation of where all these different axons are crossing each other, how they're connected. And 20 years ago or so, we would say, well, we're going to do all this in tractography. Well, 20 years later, we kind of figured out like that's a much harder nut to crack than we ever thought, because these pathways are so small, right? They're non-myelinated, small diameter. They're crossing this gigantic superhighway of white matter in the internal capsule. It's very hard to trace them. It's a mess, right? So we came up with a different approach. And a different approach was, I know that there's all these neuroanatomists out there in the world, Peter Strick, Suzanne Haber, Yolande Smith, Martin Perron, like they have a model in their head of what these pathways look like in real life, right? But they're not computer scientists, and they're not, visualization experts, right? They're hardcore anatomists. And so when these holographic head-mounted displays started, popping up from Silicon Valley, we kind of thought, well, wait a minute, one cool thing that we'd love to be able to do with these, kind of data sets is, well, we can build, kind of anatomical representations of all these different pieces and put it into the context of modern human connectome brain atlas space, right? So it's not tractography, because we're not going to use tractography, because especially at this time, like by 2020, I was like a tractography denier. Like I hated it. Okay. Kind of coming back around now, but whatever. All right. So I had gotten burned by tractography. I told everyone that it was going to be great. It was going to solve all these problems, and it created more problems than it solved. And so I became a denier. And so this was my response to it was, those little dots, right? Those dots in the orange line, like we know waypoints along the trajectory of where these things should be based off of old school histology slices that had come from human cadaver studies, for example, right? But up until this point, not many people had actually thought about, well, let's overlay these old school histology with modern, kind of human connectome brain atlases. And then we can kind of create anatomical priors of what we think these trajectories should be. And then we can kind of populate that with, kind of splines. And then we can have real anatomists come in and say, well, yeah, that kind of looks right, but it's probably wrong. So let me tell you how to do it right. And then spit out, I guess, something that you would call, like an anatomist three-dimensional consensus drawing of, these pathways in the brain. So one of the key parts of making that, kind of model was this hyperdirect pathway and its inputs into the subthalamic nucleus, right? So once again, by this time, late 2010s, we already knew the hyperdirect pathway was a key element of therapeutic deep brain stimulation. Suzanne's group had done these, elegant hyperdirect, kind of termination mapping experiments in the macaque, and come up with these kinds of somatotopic, kind of organizational patterns of the kind of the motor parts of the subthalamic nucleus and the hyperdirect innervation, and then the, kind of the prefrontal cortex parts of that. And so we could use these anatomist, kind of monkey neuroanatomy data sets in combination with, kind of our known human, kind of white matter reconstruction strategies. Once again, kind of derived from tractography, but, more like, let's start with the anatomy first and let tractography or let diffusion weighted imaging just give us, a little bit of guidance. So, on the other end of that spectrum, and we could come up with this three-dimensional map of, let's call it the hyperdirect innervation of this subthalamic nucleus. Okay, then we want to be able to put that into the context of doing computer simulations on a patient-specific level of where and what are we stimulating. Now we've moved into modern times with, these are what current clinical what they call them directional deep brain stimulation electrodes look like. So there's actually, these bands here are now broken up into 3 contacts that can stimulate, with preferential direction, anterior versus posterior versus lateral. So once again, if we put our electrode into patient space, we've got our STN, we stimulate through this contact where we'd say, okay, we know this is the hot spot right here anatomically, but what does that mean in terms of this rat's nest of literally 25 different axonal pathways converging in this one little spot? So how do we do that? You kind of, once again, you start with this anatomical fiber distribution, you apply your stimulation to that collection of axonal pathways, and then you pull out the streamlines or the pathways or the kind of axon models that are directly stimulated by this very specific stimulation parameter setting, of which you have almost infinite different parameter settings that you could simulate and test in a given patient, and then you've got literally thousands and thousands of patients, right? You could start to build up a statistical probability map of, well, which of these different pathways in the subthalamic region are really the most important for therapeutic benefit. And so in this particular simulation, even though this is just an N of 1 experiment, you get it. I picked it because it shows things that I want to tell you, which is that, with good, robust therapeutic benefit from subthalamic deep brain stimulation, you can kind of see it. There are very few of these little green lines. These green lines are the subthalamic nucleus neuron lines that go from the STN to the globus pallidus. You get some of that, but it's not really like the key player. The key players in my analyses have been this blue pathway, which is the pallidothalamic pathway. So it's going from globus pallidus, across the internal capsule, and then up into thalamus. And then this pink pathway, which we call this motor hyperdirect pathway. So it also, the axon keeps going. The parent axon is coming from layer 5 pyramidal neuron down through the internal capsule, and it keeps going down into the brainstem. But it has this collateral that shoots off and goes into and innervates the subthalamic nucleus. Now, we also, unfortunately, almost always get a little bit of stimulation of the internal capsule fibers of passage. These are these white lines here. And so that's our boundary. We're always battling, stimulating the internal capsule. You stimulate too much of that, you get motor contractions, and you now have created a side effect that's worse than the beneficial effect. So you're always kind of trying to get that balance just right. And the collection of pathways that you stimulate is highly dependent on both the electrode location, right? So if you move the electrode location, you stimulate completely different things, and your stimulation parameter settings, right? So if you use different stimulus pulse widths, you stimulate, in a very different way. So we're looking at the percent of a given pathway that's being activated by a given stimulus amplitude for, the given stimulus pulse width. One thing, though, that is very consistent in all of these simulations that we do is that the hyperdirect pathway here in pink, it is almost always the most robustly activated pathway, right? If we look at all this anatomy and all these different candidates, whether or not it's the only thing that matters is a different kind of question. But there's no doubt that if you're stimulating the subthalamic nucleus, it's almost impossible not to stimulate this pathway, right? And so once again, now this concept of anatomy converging on, a candidate of interest from the recording side of the equation, the anatomy side of the equation, and then also on this kind of stimulation side of the equation, right? So then we come to this, kind of thing. Well, all right, nowadays, everyone and their brother wants to do connectomic DBS modeling. And in my opinion, that's a mistake, because it's a whole other question and conversation. But people love this stuff right now, at least in my research world. And what they want to do is they want to quantify how stimulation in a given part of the brain translates into cortico, kind of network connectivity. And it's a super interesting question. I think it's very, very pertinent, but we also have, lots of limitations in our tools that we can actually use to analyze this kind of question. So, but let's think about, all right, if you spend enough time building models, you know that you can make a model do almost anything you want. And models are very, very dangerous things, especially to people that don't know how parameters in models impact results, right? And let me tell you, Cubby's in the audience. Cubby Gunnalan did a lot of that stuff earlier. You can make anything happen, right? And so the only thing that matters is before you start making big claims is you got to validate the model, right? Validate it with actual electrophysiology, right? It could be based on anatomy. That's one step, but you got to validate it with some other independent measurements. And so how can we do that? Well, one thing is that on the clinical side of deep brain stimulation research, people have gotten really excited recently about what we call these cortical evoked potentials. So we stimulate in one part of the brain, and then we've got electrodes in some other part of the brain. We record there, you get an evoked potential. Now you've got a measurement of that this part of the brain is connected to that other part of the brain, right? And so this is an example of work that Svetlana Miakinovic was doing at UCSF at the time and Emory now, where as part of the deep brain stimulation surgery, they would put a temporary cortical strip electrode over sensory and motor cortex. And then down here, you can't see it in the picture, but they would also be stimulating in the subthalamic nucleus. And then they would record in cortex, you get your stimulus artifact. And then in about 2 to 3 milliseconds after the stimulus, you get evoked potential 0. And then about 3 or 4 milliseconds, you get evoked potential 1. And then at about 5 seconds or milliseconds, you get this next one, right? So you got these signals, very robust, reproducible electrophysiological signals tagged to your stimulus. All right. So then, it's like, well, we can actually dissect the biophysics of those cortical evoked potentials using these kinds of models, right? And as a validation check that the model actually is lining up with something real. So once again, same kind of thing. You've got your hyperdirect pathway in pink. You've got your motor internal capsule in white. One of these is quote unquote therapeutic. The other is side effect. We apply in this case, they almost always use bipolar stimulation for these experiments because it really limits your stimulus artifact. And there's other benefits of that I'll get to later. You get then a prediction of how much of these different pathways you think you're stimulating with the given stimulation parameter settings. And then now we created action potentials in this fiber, and then we can measure how long it takes that action potential to go up to the layer 5 pyramidal neuron, right? We can look at the conduction delay. You run that simulation, and in this blue axon or this blue action potential, boom, this would be what we would call an internal capsule, large internal capsule fibers of passage, 10, 15 microns in size. That's going to get up to cortex right at exactly the time that this first little bump in the evoked potential arrives. And then if we then look at now the next thing over, what if we had a hyperdirect axon that was pretty big but not giant? That one would line up almost perfect with the second hump, and then a hyperdirect axon, but it was a pretty small diameter fiber. That one would line up with this third hump, and so on and so forth, right? So that you could actually dissect the biophysics of where these evoked potential electrophysiology signals are coming from. And if you then do that with many, many, many stimulation parameter settings, right? Because you run these experiments in several patients, and then in the operating room, you go through all kinds of different settings, all kinds of different electrode configurations, different stimulus amplitudes, different stimulus pulse widths, right? And then you look at, does our prediction of activation of the hyperdirect pathway line up with the amplitude of the evoked potential wave that we know biophysically should be related to that signal, right? You go through that kind of process and like, well, that's pretty good correlation. Like, I don't know, 0.8 or something. Like, never in the history of neurostimulation modeling has anyone been able to come close to that kind of thing, right? So we're like, we're on the right track. We're starting to figure out how to make these models both anatomically realistic, electrically realistic, and physiologically predictive of something that can be really measured in real life. So that was pretty cool. But